Good evening, everyone. I am Amrita, PhD scholar from University of Hyderabad, and I take live interactive sessions for animal physiology every Tuesday from 7 p.m. to 9 p.m. So today we are in our week 9 of this course, and we have successfully discussed 8 weeks of previous year questions as a part of these interactive sessions, and we discussed multiple questions throughout these 8 weeks related to the course Animal Physiology, right? So every week we discuss around 10 questions of that particular week given in the previous year so that you all can revise the concept again through the questions. You can know how much knowledge you have gained through the lecture of that particular week, and you will have convenience in solving the current year assignments. You all must be getting current year assignments, and if you solve these questions, it will give you an idea of about how to solve those questions as well, right? So we have completed 8 weeks. All the classes are recorded and are uploaded in my YouTube channel. Today we are in our week 9. So today again we are going to discuss 10 questions about the neural system, and today will be the last week we will be discussing about the neural system. Next week we will be discussing another topic about in animal physiology, right? So I think it is already 7:15, and if we have waited enough, whoever has joined the live interactive session, fine, and whoever is watching the recording sessions, this session is for both of them. So the rule of the class is that I will be showing you the question. I'll give you 1 to 2 minutes time to think which can be the correct choice for that question, and you need to write it in the chat box. Whoever is attending the live session, they need to write it in the chat box, and whoever is seeing the recorded session, they can just note it down somewhere. Okay. So after 1 to 2 minutes, I will show you what is the correct answer to that question, and we will discuss an correct explanation for that option as well. Okay. So I think we shouldn't waste more time, and we will start with the first question of today's session. Okay. So the first question is, pheromones are sensed by which sense of ours? Pheromones are sensed by which sense of ours? Auditory, olfactory, vision, haptic. Okay, I already got the answer of the student who has joined through this live session. Still, I'll wait for 2 minutes so that whoever is seeing the recording, they can also have a practice like this. Okay. Okay. I think that much is enough time. It is a very simple and direct question, I think no complications in solving this question at all. So the correct answer is olfactory. So pheromones are sensed by the olfactory receptors, or you can say the olfactory senses. So what sense detects pheromones? So particularly in mammals, pheromones are detected by the vemeronasal organ called as VNO, abbreviated as VNO, a bilaterally symmetrical cylindrical organ. You can see here encased in a bony capsule on the anterior nasal septum. VNO neurons send a single unbranched axon to the accessory olfactory bulb. You can see the olfactory tract here. This is the olfactory bulb, and which in turn sends projections to the verumonasal amygdala. Okay, so here you can see the poorer layering of olfactory sense, the olfactory tract, the olfactory bulb, the connective tissue, and the olfactory epithelium, which contain these olfactory receptor cells. Okay, okay. We'll move on to the next question. Which of the following is not a cell type found in human eye? Which of the following is not a cell type found in human eye? Bipolar cells, ganglion cells, amacrine cells, or goblet cells? Start answering. Okay, done. Let's see what is the correct answer to this question. The correct answer is D. So, goblet cells is the type of cell which is not found in the human eye. But where is it found then? If it is not found in the human eye, where it is found? So, goblet cells are a type of intestinal mucosal epithelial cells, and the primary function of these goblet cells is to synthesize and secrete mucus. So, they are a part of the intestinal lining, right? They are not a part of the eye. In eyes, we can see the amacrine cells, the bipolar cells, right, and the ganglion cells as well. Okay, clear? We'll move on to the next question. The photoreceptor cell rods detect intensity of light, color of light, both A and B, and none of the above. Start answering. A very simple and easy question again. I'll wait for more 1 minute before we discuss these questions. Whoever is joining late, they can start. We can continue from the third question. Okay. Okay. Done. We'll see what is the correct answer. So the correct answer is A. The photoreceptor cell rods detect the intensity of light. We will discuss the explanation for this question after you all answer the fourth question, which is the photoreceptor cell cones detect intensity of light, color of light, both A and B, or none of the above. Start answering this question. Then we'll move on to the explanation of answers for both these questions. Okay. You're unable to see the screen, but I think the other student, Khushbu, she can see the screen. She's answering the questions. So I don't think it is a problem from my end. Please check from your side. Okay, I think I have given enough time. This was a very simple question, and the answer was very obvious. the other option. The photoreceptor cell cones detect color of light. So the photoreceptor cell, which are rods, they detect the intensity of light, whereas cones are responsible for detecting the color of the light. Right? So what are rods and cones? They are the receptors in the retina responsible for your sense of light. They are the part of the eye responsible for converting the light that enters your eye into electrical signals that can be decoded by the vision processing center of the brain. So, cones are responsible for color vision, whereas rods sense the intensity of the light. Here you can see the wavelength of the light that rods and cones detect. While for rods, there is only a single black line because it sense the intensity in this range, whereas there are multiple colored lines which blue, red and green light line under this wavelength of range of wavelength, which is detected by the cones. And here you can see how rods and cones are different in structure, how they look like. Okay. Okay, we'll move on to the next question. Fifth question. During an eye transplant, which is transplanted or replaced? Whole eye, retina, cornea or lens? Very simple question. Start answering. Okay, done. Let's see what is the correct answer to this question. It is cornea. So during an eye transplant, what is generally transplanted with, that is the cornea, because eye transplant is done when your cornea is damaged, which is responsible for your vision, and hence cornea is transplanted during an eye transplant. Okay, we move on to the next question. Sixth. Cataract leads to dash, and during surgery it is replaced by dash. A. Blood cornea, artificial cornea. B. Cloudiness of lens, intraocular lens. C. Retina damage, intrarenal retina. And D. Pupil dilation or Botox. Read the question carefully and answer. Cataract is a very famous disease related to the eye, and I think everyone should know what causes it and how does it look like. Okay, it is a very common disease as well. Okay, everyone done? Let's see what is the correct answer to this question. The correct answer is B. So, cataract leads to cloudiness of eyes, and during surgery, it is replaced by intraocular lens. So, here you can see how a normal eye and a normal lens looks like, and how a eye suffering from cataract looks like. A cloudy layer, a one white layer is formed in front of the pupil, known as the clouded lens. Another lens forms, which is the clouded lens, and it damages your vision. So for that you need a surgery to correct this cataract, and in this surgery this cloudy lens is removed and an intraocular lens is kept in place of it. Okay, so we'll move on to the next question, which is the seventh question. At the blind spot there are dash photoreceptors: maximum, no, half the density of, horizontally aligned. Very easy question again. I think everyone can answer it. Okay, everyone done? Let's see what is the correct answer to this question. The correct answer is B. As the name suggests, at the blind spot there are no photoreceptors. That's why it called a blind spot. So it is outside the center of the vision, and in this place where there is a blind spot, there are no rods and cones, which are the photoreceptors, right? Okay. We'll move on to the next question. Eighth, dark current is depolarizing, hyperpolarizing, either A or B based on the color of the light, either A or B based on the intensity of light. Okay, let's see what is the correct answer to this question. The correct answer is A. Dark current is depolarizing. So here you can see dark current, which is known as the depolarizing current. Why it is known as the depolarizing current? Because it is carried by the sodium ions that flows into a photoreceptor cell when unstimulated. So in the presence of darkness, what happens? There is a depolarization, that is sodium ions get carried into the photoreceptor cells when it is unstimulated. You can see how the signaling pathway works in the eye during darkness and during the light conditions in the presence of the light. So it is depolarizing in the darkness. That's why it is called as depolarizing current. That is, it is in negative. That's why. Okay, we'll move on to the next question. Resting potential for photoreceptors is minus 90 millivolt, minus 40 millivolt, minus 75 millivolt, or plus 90 millivolts. Start answering. Okay, let's see what is the correct answer to this question. The correct answer is B, minus 40 millivolts. So here you can see again the same thing. What happens in the presence of darkness and what happens in the presence of lightness. So when the environment is dark, there is maximum glutamate release, and when the environment is light, there is reduced glutamate release. Depending on that, the resting membrane potential of the photoreceptors changes to minus 40 during darkness, and during light it decreases to minus 60 millivolts because of the reduced Na plus in through the CGN channels. Because in darkness the Na ions come into the cells cross the channels, whereas in case of light they go out of the cells, which leads go out of the channels, which lead to the reduced sodium ion concentration, leading to more negative charge. Whereas in case of darkness, the sodium ions go through the CGN channels and come inside, which leads to an increase in the membrane potential to minus 40 millivolts, which is the resting membrane potential. Okay, we'll move on to the last question of this session. Which part of the eye helps in changing focal length of the lens? Retina, pupil, cornea, or ciliary muscles? Very easy question, basic question. We have all learnt in 10th standard, 9th, 10th standard, right? Okay, let's see what is the correct answer to this question. The correct answer is ciliary muscles. So ciliary muscles is the part of the eye which help in changing the focal length of the eye. So how the eye focuses the light? With the help of the ciliary muscles. So when it has to focus at a distance range, the ciliary muscles are relaxed. Whereas when the eye has to focus in a close range, the ciliary muscles get contracted and change the focus so that you can visualize properly. Okay, so the change in focal length of the lens is dependent on the relaxation and contraction of the ciliary muscles. Okay, with this we are at the end of our today's live interactive session. We discussed 10 questions on eye basically, and I think those were very easy questions, and who have attended the live session, I was seeing their answers. I think everyone has given the correct answer, which is wonderful. And whoever is practicing through the recorded sessions, I hope these questions are helpful to you guys, and please revise these questions, the lectures, the previous live interactive sessions. All of these are very important from your exam point of view, as you will be getting multiple choice questions as well as subjective questions. Okay, so solve your current assignments properly because they carry marks, as well as keep practicing these previous year questions as well. If anyone has no doubts, we can end the session. Does anyone have any doubts, or should we disperse? Okay, no doubts? Great. Okay, we'll stop the session today here, and again we'll meet next Tuesday with the new 10 set of questions for week 10. We have 3 more weeks left, week 10, 11 and 12, and I hope that will go smoothly as well like all the previous weeks. And yes, we'll be discussing with some new topics in the next session. Till then, keep learning. Okay, thank you everyone.